Hello everyone. I am Dr. Pooja Johri, your original guru for biochemistry. Now, all of you must be preparing for your INI CET for the coming year. So, this current video is actually I will be discussing a very important, examination point of view questions which have been asked in previous years of INI CET. And I will be discussing some of the important core areas from which questions can be asked. So guys, let's start up with the discussion. Now see, first of all, let's see question number 1. Vitamin A toxicity will cause damage to which of these? Now, this is a very important question. As you all know the pattern of the questions, most of the questions have been asked on vitamins. Now, this question is about vitamin A. Now, we all know. Now, before giving the answer to this question, let me just tell you what are the important points which can be asked on vitamin A. The most important feature of this is actually this vitamin is a fat soluble vitamin. And always remember, whenever we talk about fat soluble vitamins, they are stored. And fat soluble means they are insoluble in water and they will always require a carrier protein for their transport. And the active forms of vitamin A are retinol, retinal, and retinoic acid. And the interesting part about these three forms is they are interconvertible with respect to one another. Be it alcohol, be it an aldehyde, be it an acid. All these forms are interconvertible with respect to one another, and all three have a different role to perform. And this vitamin A is actually has a very important role to play in vision. So if we talk about retinal, it has a role to play in vision. If we talk about retinol, it has a role to play in reproduction. And if we talk about retinoic acid, it plays a role in cell growth and differentiation. Now, coming over all the three forms, just now I have highlighted the important functions of this. Now see, always remember vitamins play a very important role in normal functioning of the body, right? And obviously they are not synthesized. Some of the vitamins are synthesized, so they have to be taken from outside diet. Now there has to be a recommended dietary allowance for all the vitamins. Now, what is the significance of this recommended dietary allowance? Means it has to be taken in a dosage, prescribed dosage. If we increase the intake of vitamin, it will lead to hypervitaminosis, which is equally harmful. And if we decrease the dosage, it will be known as hypovitaminosis. Now, coming over to the question says vitamin A toxicity. That means if we increase the intake of vitamin A, which organelle will be damaged the most? So, the answer to this is lysosomes. Because always remember, if we have to differentiate between a fat soluble vitamin and a water soluble vitamin, all the fat soluble vitamins in general are stored in the body. And if we talk about water soluble money, they are soluble in water, so they are excreted in urine as such, except B12. Right? So in vitamin A, they are stored, and this vitamin A is stored in the liver. But if the concentration of vitamin A increases, it will lead to hypervitaminosis, and the damaged organelle will be lysosomes, because we know that lysosomes are also known as the suicidal bags of the cell, because they consist of the enzyme hydrolases. Right? Now, what are the features when hypervitaminosis occur. So we can say damaged organelle, lysosomes, right? And since vitamin A is stored in the liver, so those people who stick to their diet is this, bare, they are more likely to be, prone to this hypervitaminosis. And it leads to what? The clinical features include alopecia is there, then dry pruritic skin, right? Then hypervitaminosis can lead to pseudotumoral pseudotumor cerebri, and then it can lead to increased concentration of TGs leading to hyperlipidemia. It can lead to hepatomegaly, enlargement of the liver. So these are the clinical features when the increase in the concentration of vitamin E takes place or vitamin A toxicity leads to these. So the answer to this question is lysosomes. Then moving on to the second question, magenta tongue is seen in the deficiency of. This is again a very, very simple question and a very important question. Vitamins is a very, very important topic on which roughly every year questions have been asked. So if we talk about this, magenta color is it is seen in the deficiency of vitamin B2, whose active form is riboflavin. Now coming on to the options, pyridoxine is simply B6. Active form of pyridoxine is PLP. Riboflavin is B2. Active form is FMN. Pantothenic acid is B5, whose active form is SH-CoA. And niacin is vitamin B3, whose active form is NAD. So first of all, let's discuss the question. Magenta color, the answer is riboflavin. Now, what is this riboflavin? First of all, it is a water-soluble vitamin. And always remember that all the B complex vitamins function as coenzymes. And this is again one of the very, very important point of difference between fat soluble and water soluble. Fat soluble में only vitamin K functions as a coenzyme, whereas in water soluble B complex, all of them are functioning as coenzymes. So this is again B2 riboflavin, whose active form is FAD, flavin adenine dinucleotide, and flavin mononucleotide. And you have seen, you must be familiarized with so many, be it carb metabolism, be it amino acid metabolism, FMN or FAD as used as coenzymes. Right? Then functions, again it is functioning as, if I write the function will be coenzyme. And if we talk about the deficiency manifestations, the most important are characterized by, first of all, two C's. This is again one of the very important mnemonic. This is how we can write. Two C's means corneal vascularization, and second C stands for chelosis. Right? And other deficiency manifestations, apart from corneal vascularization and chelosis, it leads to angular stomatitis, then glossitis, right, and seborrheic dermatitis. So we can say that vitamin B2 or riboflavin deficiency manifestation is mainly 5. 1, 2, 3, 4 and 5. Corneal vascularization is there. There is increased blood flow leading to pink color to the eye. Then cheilosis is there. Then angular stomatitis is there. Glossitis is there, which means magenta color of the tongue. Okay, there are fissures at this junction and obviously seborrheic dermatitis is there. So these are the deficiency manifestations of vitamin B2, which is a riboflavin, and the active form is FAD and FMN. If we talk about niacin, this is also a very important question from examination point of view. Niacin deficiency leads to pellagra. And we all know this pellagra is characterized by 3 Ds. Diarrhea, dementia, and dermatitis, which can lead to the 4th D, which is death. And niacin is a very important endogenously synthesized vitamin also. We all know that 60 milligram of tryptophan, this is again a very important question. 60 milligram of tryptophan is required to synthesize 1 milligram of niacin. So we can see in one shot we are able to solve 2 questions. And this niacin deficiency is characterized by Kesel necklace also. This is also one of the important features, right? So this is the answer to question number 2. Then moving on to question number 3, that all these are true about collagen except. Let me read out all the options first. Phenylalanine is the most abundant amino acid forming it. Answer to this question will be this only, but let me read the other options also. It has a triple helical structure. Yes. It is the most abundant fibrous structure of fibrous tissue. Correct. It is formed by lysyl and prolyl hydroxylation to enhance the stability of the structure. So all the 3 options of collagen are correct. Right? Now, first of all, what is this collagen? Now, all of us know that collagen is the most abundant connective tissue. And collagen has a triple helical structure. Helical structure. Triple helical, and it always forms 3 repeats of the amino acids. Now, what are the 3 amino acids which are forming the entire structure of collagen? Glycine, it has repeats of glycine, proline and lysine. Always remember, collagen, which is the most abundant connective tissue, it has 3 amino acids: glycine, proline and lysine. And a very important phenomenon takes place. It is present in an inactive form. It is converted into its active form. Means there is post-translational modification. Because all of us know that translation, or there is synthesis. There is a central dogma of information, means DNA forms RNA and RNA translates the information which is already hidden in the form of genetic code. So post-translational modification of this takes place. And what happens is vitamin C is required and the process is known as hydroxylation. Hydroxylation means attachment of hydroxyl group. So proline and lysine undergoes hydroxylation and it forms, the enzyme is prolyl hydroxylase, which converts this proline into hydroxyl proline, and lysine in the presence of lysyl hydroxylase to form hydroxyl lysine. So it is giving more strength. That is why we say if there is a deficiency of this vitamin C, it leads to defective. I'll just write it here. Vitamin C deficiency leads to defective collagen formation. And if collagen formation is defective, it leads to scurvy. The reason behind this is this only, right? And if we come over to, this is how collagen is synthesized. Okay, you can say glycine, proline, lysine, they undergo hydroxylation to form prolyl hydroxylase and lysyl hydroxylase. And if you look at the options, you can see that all are coming. It has a triple helical structure formed by this, formed by this. Coming over to phenylalanine, phenylalanine is altogether a different amino acid. We all know that phenylalanine is a essential amino acid. Why essential? Because it is required for the synthesis of tyrosine. Tyrosine, which is required for the synthesis of melanin in the body. Similarly, it is required for the synthesis of catecholamines, epinephrine and norepinephrine. And similarly, tyrosine is required for the synthesis of T3 and T4. So phenylalanine is an amino acid which is essential, but it is required for the synthesis of tyrosine. It has nothing to do with collagen. So the answer is this. Except phenylalanine, all the features are of collagen. Then moving on to question number 4. Sequence of PCR. Now this is also a very important question. PCR, we know that it stands for polymerase chain reaction. Played a very, very important role during the time of COVID. So what is this PCR? If we look at this, a very important molecular biology technique which has, solved so many important, which has so many important features. So we won't take it into that much detail, but the basis of PCR will be the same. Because if we see, there are so many types of PCRs. RT-PCR is there, nested PCR is there, anchored PCR is there, fluorescent PCR is there. But the basis, the prerequisites of PCR remains the same. And what is it? It is simply, how do we define it? It is exponential amplification. Of DNA. And it is 2 raised to power n. It runs at a very high temperature. So 2 raised to power n means if PCR runs for 2 cycles, so how many amplicons will be produced? Now if you look at the options, or you can say what are the prerequisites? The prerequisites of PCR, it always requires an enzyme which is thermostable, because PCR always runs at a very high temperature. The temperature is like 80 to 100 degrees. So enzyme which is actually thermostable. And the enzyme used is DNA polymerase. It requires the primers because actually it is the hybridization, and it requires magnesium ions, right? And obviously it requires DNA, or you can say DNTPs. I'll just write as NTPs. These are the prerequisites to run any PCR. Ab if it is RT-PCR, so RNA will be converted into cDNA. That is what is reverse transcriptase all about. So these are the prerequisites. Up coming over to the steps of PCR, what are the main steps? So the main steps to run a PCR are, first of all, DNA strand is denatured. So we denature it because denaturation, because the two strands which are held together by hydrogen bonds, the temperature is raised and the hydrogen bonds are broken because we have to set the DNA strands apart. Then second is renaturation. Renaturation, or you can say the most appropriate would be annealing. Because annealing or renaturation mein what is the difference? That D means removal of the naturally existing state of DNA. Re means we are reuniting DNA strands. Annealing, when we slowly try to bring the temperature down. Because when we are increasing the temperature and when we immediately decrease the temperature, there are chances that DNA strands might not join also. So annealing is slowly cooling, but a slow cooling of DNA. So after denaturation, annealing is done and then extension is done. Extension, why? Because we are adding the primers. So primers, as per their complementary base sequence, they will try to hybridize. And once extension, when the amplicons are amplified, once we know hybridized, or we can say, you can say the amplicons, or you can say the DNA strand starts reuniting, then termination is there. Now the PCR cycle needs to be stopped. Now coming over to the answer, these are the main steps. So we will see the order is the same. Denaturation, annealing, extension, and then termination. We'll look at the options. Answer will be A. You got my point. First of all, the two strands are denatured, the hydrogen bonds are broken. Then we try to slowly cool down the temperature by adding the primers. Primers are designed already. And then once it undergoes hybridization, there is complementary base pairing, then in the end, it is terminated. Right? So this is the sequence of PCR. And what is the significance of PCR? PCR is carried out to check the disputed parentage. These are the applications. To check the different type of aneuploidy also. Like this. Then moving on to question number 5. Deficiency of which vitamin leads to accumulation of methyl tetrahydrofolate and decreases efficiency of functional folic acid? Now this is a little bit, let's say it is a very easy question, but little bit complication is there. See, what is this methyl tetrahydrofolate? We know that it is what? It is the active form of folic acid. And the question is saying it is decreasing also the efficiency of functional folic acid. So just try to recall, I have mentioned it in my videos also, there is one reaction in the body in which B9 folic acid and B12 cobalamin are interdependent on one another. That reaction is in the metabolism of methionine. If you look at the options, B12, folic acid, vitamin C and B6. Let me just discuss the reaction and then we will come over to the options. See, what happens is the reaction is this. What happens is This methionine, we know that it is an active donor of methyl group. Before it donates a methyl group, methionine is activated to form S-adenosyl methionine. This S-adenosyl methionine donates its methyl group to the acceptor and itself is converted into S-adenosyl homocysteine. This S-adenosyl homocysteine then donates this ATP because now its function is over and it will give this adenosyl group and it will be converted into homocysteine. Now here lies the main concept that this homocysteine needs to be converted into methionine. Right. Moving on to question number 5. Deficiency of which vitamin leads to accumulation of methyl tetrahydrofolate and decreases the efficiency of functional folic acid? Now see, this I have discussed in my videos also. Just try to recall one reaction in the body in which folic acid and which is B9 obviously, it is dependent on one very important, I should say, co-vitamin which is B12 cobalamin. There is only one reaction in the body in which B9 and B12 are interdependent on one another, which is related to the metabolism of methionine. Now, if you look at the, first of all, options may B12 is also given, folic acid is also given. So, obviously, you guys might be confused ki would be the correct answer, whether it is folic acid or B12. So, let's see the explanation first of all, then we will come over to the answer, right? So, here just remember, just try to recall one thing that methionine. Methionine, which is a sulfur-containing amino acid, a essential amino acid, it is required for transmethylation reactions in the body. Okay, so this methionine, before it donates a methyl group, it needs to be activated. Obviously, activation के लिए it requires ATP. So it forms S-adenosyl methionine, which is SAM. This S-adenosyl methionine donates this methyl group to the acceptor and is converted into S-adenosyl homocysteine. Okay. Now this S-adenosyl homocysteine, once the function is over, क्या करेगा? It will simply give this adenosyl group, function over, काम खत्म, it will give that ATP and it will be converted into homocysteine. Now, if we look at the structures of methionine and homocysteine, there is only a difference of the methyl group. Now, here the interesting part is from where this methyl group is coming. Now, this methyl group is coming from, first of all, folic acid, and B12 is helping in the reaction. So, this reaction, I can put it like this, that methyl B12, once it donates this methyl group, it will be converted into B12 and this reaction is linked to methyl THF and THF. THF is tetrahydrofolate or B9, which is the active form of folic acid. Now see, here the question is what? Deficiency of which vitamin, okay, leads to accumulation of methyl tetrahydrofolate. Now see, if I say B12 is absent, this is absent. Means the donor, or you can say the carrier, donor is folic acid. Folic acid is handovering this methyl group to cobalamin so that it can donate, it can give this to homocysteine to form methionine. If this B12 is deficient, that means this reaction cannot take place. If this reaction cannot take place, it will lead to accumulation of this methyl group. And then what will happen if this methyl group increases, the concentration increases? We call this term as folate trap. Means folic acid is trapped by the deficiency of B12. And that is what is here. Deficiency of which vitamin leads to accumulation of methyl THF? It has to be B12. So the answer will be B12. A option. Not folic acid, because the accumulation kiski ho rahi hai? Accumulation is at the part of this because B12 is absent. And this is one of the very, very important feature of this also. This is actually known as non-classic homocystinuria 2. Homocystinuria, where there is increased concentration of homocysteine and decreased concentration of methionine. Right? So the answer is clear-cut B12. Moving on to question number 6. Thiamine deficiency causes lactic acidosis due to the dysfunction of which enzyme? Now see, this question is a very, very important question. Why it is important? Because this can be asked in so many other different ways also. What is the significance of this thiamine? What is lactic acidosis? These are the important uses of this. Now see, coming over, we'll take each and everything one by one. First of all, try to understand this reaction is otherwise important. What happens is, all of us know that thiamine is vitamin B1. And we know the active form of B1 is TPP. Now, TPP is, just now I told you, when I discussed question number 1 and 2, I told you that most of the B complex vitamins function as coenzymes. So this B1 acts as a very, very important coenzyme for pyruvate dehydrogenase enzyme. Coming over to the function of this, what happens is pyruvic acid, we know that pyruvic acid is coming from the anaerobic and aerobic breakdown of glucose, isn't it, by glycolysis. And this pyruvic acid, then what is the fate? That if it enters in the mitochondria in the presence of oxygen, it loses one molecule of carbon dioxide, forming acetyl coenzyme A. In the absence of oxygen, it forms, which happens in the skeletal muscle, it is converted into lactate. Now, what is the difference between these two reactions? This reaction is a reversible reaction, and this reaction, as you can see, it is a irreversible reaction. If we come over to the enzymes, this is PDH and this is LDH. We all know LDH is lactate dehydrogenase. Right? We all know PDH is pyruvate dehydrogenase. Plain and simple, right? Now, what is the difference between LDH and PDH? One reaction, it is clear. LDH is catalyzing a reversible reaction. PDH is catalyzing a irreversible reaction. Coming over to this, thymine is a component of PDH. And you know, this pyruvate dehydrogenase is an example of multi-enzyme complex. Now, what do you mean by multi-enzyme complex? This PDH requires B1, B2, B3, B5 and lipoic acid. And 3 enzymes. And you see B1, this is the most important one because it has 3 carbon atoms, it has 2 carbon atoms. One molecule of carbon dioxide is taken over, means decarboxylation. May the main coenzyme is TPP. Now coming over to the question, let me just take you to the question. Question is, Thymine deficiency causes lactic acidosis, which means if thymine is absent, this reaction will not take place. So obviously pyruvic acid का concentration बढ़ जाएगा. If the concentration of pyruvic acid increases, concentration of lactic acid will increase. Clear? So we can say lactic acidosis हो जाएगा. Now see, due to the dysfunction of which enzyme? It has to be pyruvate dehydrogenase, because TPP is a coenzyme for pyruvate dehydrogenase. अगर हम बात करें पेप कार्बोक्सीकाइनेस is an enzyme of gluconeogenesis, right? Pyruvate decarboxylation करेगा, but it is not an enzyme for dehydrogenase. Transketolase, it is an enzyme of HMP shunt. So here you can say the answer will be PDH or pyruvate dehydrogenase, which is a very, very important enzyme of multi-enzyme complex. अब इसमें एक और question आता है कि that the you can say the coenzyme or vitamin required for cellular respiration, because this reaction which I listed, it is a link reaction. Why it is a link reaction? Because it links glycolysis and TCA, because it takes place in the mitochondria. And we all know that TCA plays a very, very important role, being an amphibolic pathway, or you can say it gives energy to the brain. So the answer is PDH. Then moving on to question number 7, Wobble hypothesis proves which nature of codon? A very simple question and a very, very important question from examination point of view. Wobble mane wobbling mane change karna. Wobbling hypothesis means degeneracy of the base pair. Now what do you mean by degeneracy? If I say U is there, if I say UUA is there, if I say UUG is there, what will happen is change in the third base pair. Change in the third base pair is actually degeneracy, which is also known as wobble hypothesis. Wobbling karna, because it will not make any significant change. अब reason इसके पीछे क्या है? Because we all know that genetic code is triplet, isn't it? This is the first important property. Total हमारे पास कितने amino acids हैं? We have only 20 standard amino acids. So we have to code only for 20 standard amino acid. It is triplet. So obviously what will happen is कि third base pair might change, and we can say that these three you can say codes might pair for one single amino acid. This is what is degeneracy or wobble hypothesis. Then moving on to the 8th question, which of the following is not glucogenic substrate? Again, a very important question. I remember some 3-4 years back, there were 2 questions in a exam only which were asked on gluconeogenesis. Very, very important. First of all, how do we define gluconeogenesis? Gluconeogenesis. Simple. Genesis means synthesis. Synthesis of glucose. कहाँ से? From non-carbohydrate precursors. This is the definition of gluconeogenesis. Synthesis of glucose from non-carbohydrate precursors. Now, what are the other non-carbohydrate precursors? Alanine, lactate, glycerol, propionate. These are the four very, very important non-carbohydrate gluconeogenetic substrates. Then gluconeogenesis occurs in two organelles. कौन कौन से? Mitochondria and cytoplasm. One more reaction takes place, which takes place in the endoplasmic reticulum. Mitochondria mein there is only one enzyme, which is pyruvate carboxylase. This was the question which was asked. Pyruvate carboxylase is the only enzyme, pyruvate carboxylase, only enzyme of gluconeogenesis present in the mitochondria. The rest are in the cytosol. Okay? And endoplasmic reticulum mein last enzyme in which glucose 6-phosphatase is there, which converts glucose 6-phosphate to glucose. Right? So these are important points. Then coming over to the question, acetyl coenzyme mein, first of all, see, lactate is a substrate, just now I told you. Correct. Pyruvate, yes, it is also a substrate, pyruvate carboxylase, correct. Oxaloacetic acid, again, it can be there. Up, not glucogenic, acetyl coenzyme A. Always remember one thing, that carbs can be converted to fats. Always remember, carbohydrates can be converted to fats. Fats cannot be converted to carbohydrates. Because if you remember one, very important gluconeogenesis ka first step, if you remember, in which this pyruvic acid is converted into oxaloacetic acid, and this oxaloacetic acid is then converted into pyruvic acid. Here acetyl coenzyme is just acting as a modulator. Acetyl coenzyme A can never serve as a purpose, yeah, it cannot be acted as a substrate to give rise to glucose. Always remember this. Okay? So the answer will be acetyl coenzyme A. And just remember this point, this is very, very important. Carbs can be converted to fats, but fats cannot be converted. That is why whenever we say we take excess of carbohydrates, glucose will be converted into acetyl coenzyme A and to fats. But fats can be broken to acetyl coenzyme A, but directly acetyl coenzyme A can never ever form glucose. Then moving on to question number 9. What disease causes problem in collagen maturation? A very simple question, just now I discussed. Collagen maturation ke liye always remember, this is post-translational modification, and the vitamin which is required is vitamin C. Or deficiency of vitamin C leads to scurvy. So the answer is scurvy. And the reason also I told you, because vitamin C is required for hydroxylation. Kiski hydroxylation? Proline and lysine. So the answer is scurvy. Moving on to the last question, functions of different compounds of vitamin A. Now this also I discussed that vitamin A has 3 forms, retinal, सबसे पहले retinol, then retinal and retinoic acid. I told you all the 3 forms are interconvertible with respect to one another. ठीक है? Up coming over to the functions, this also I told you, retinal, whenever we talk about retinal, al means it is for vision. Al is for vision. Ol is for reproduction. Oic acid is for cell growth and development, and the richest source of vitamin A are carrots, beta carotene, which is an antioxidant. Now look at the options. A may you can say 3, B will be 1, C will be 4, so the option will be this. A की option number is 3, B का option is 1, C का option is 4, and D का option is 2. Right? So the answer will be this. So these are some of the important previous year's questions which have been discussed in your INI CET exam. And I have tried to discuss each and every possible aspect related to these questions also. So this is, I think, enough regarding these 10 questions, and I hope all the you can say solutions to these questions are now clear to all of you. So guys, wishing you all the best for your coming exam. Thank you so much.